Can we actually beat nature at its own game? Welcome to this explainer. Think about it for a second. Nature has had 4 billion years of biological evolution to perfect its designs. It is the ultimate engineer, right? But it's also incredibly slow. So what if we could just grab the steering wheel and do in a matter of days what takes nature literal eons?
And that brings us to the core theme of today's explainer, the evolution of evolution. We're looking at how scientists are no longer just sitting back and studying nature's catalysts. No, they're actively designing and engineering completely artificial, new-to-nature enzymes. It's this absolutely thrilling frontier where biology slams right into computational chemistry.
Specifically, we're going to explore what happens when we teach old proteins entirely new tricks. By taking these ancient biological building blocks and totally repurposing them, we're unlocking chemical reactions that have literally never existed in the natural world before. How cool is that?
So here's our roadmap for today. We'll start by looking at nature's chemical catalysts, then move into speeding up evolution, computational enzyme design, building artificial enzymes, and finally wrap up with the future of chemistry. Let's dive in.
All right, part 1, nature's chemical catalysts, the baseline of biology.
Now, the enzymes we find out in nature are absolutely incredible, for sure. They're these highly efficient little catalysts that basically make life as we know it possible. But they have a pretty massive fundamental limitation. They really only evolved to perform the specific reactions necessary for an organism to survive and reproduce. So, nature's catalysts, they're perfect if you need to digest food or build a cell wall. But they definitely haven't evolved to do the highly complex, super-specific synthetic reactions we need for modern industrial chemistry, things like manufacturing pharmaceuticals or clean biofuels. Basically, they're boxed in by the strict limits of natural selection.
Which brings us to part 2, speeding up evolution, taking the reins.
This right here is where things get wild. Frances Arnold actually won a Nobel Prize for exactly this shift, a process called directed evolution. See, instead of just waiting around for random mutations in nature to slowly adapt to an environment over millions of years, directed evolution condenses all of that natural selection into mere days, right inside a laboratory test tube. It's essentially a man-made shortcut built on some seriously deep molecular insights.
But where did this whole idea actually start? Well, to get the history right, we have to look back to 1984, to a theoretical paper by Manfred Eigen. He was the first to propose this step-by-step, iterative, evolutionary machine to optimize enzymes. He figured out that trying to find rare, improved protein variants in just massive, completely random libraries, that'd be almost impossible. So instead, he mapped out a workflow that used smaller libraries and multiple, carefully controlled generations of mutations.
And what's so fascinating is how Eigen's theory is put into practice today by basically forcing evolution. You start with an enzyme that shows just a tiny hint of the activity you want, something called catalytic promiscuity. Then, you throw in mutations using genetic tricks like DNA shuffling, and you apply intense selection pressure. Frances Arnold, for instance, forced enzymes to survive in highly unnatural, super harsh organic solvents like DMF. It's brutal, but only the mutated survivors make it to the next round.
So the absolutely crucial takeaway here is this rapid, iterative loop. You identify a starting protein, mutate its gene, screen for the absolute best performers, and then repeat the whole cycle. It's exactly like the selective breeding we humans have done with crops and dogs for thousands of years, but we're doing it to molecules. And through this loop, an enzyme's activity can be boosted hundreds, sometimes thousands of times over.
Moving right along to part 3, computational enzyme design, the digital boost.
Okay, let's compare these two really powerful methods side by side. On one hand, directed evolution has an incredibly high success rate, but it inherently needs a natural baseline to get started. You have to have a starting point. On the other hand, the computational inside-out design approach, that lets us dream up entirely novel catalysts totally from scratch on a computer. The catch there, though, is that their actual kinetic speed often falls pretty short on its own.
But the real magic happens when we bring them together. When researchers combine that rational digital design with the biological optimization of directed evolution, the results are just staggering. We can create enzymes with vastly improved binding, way faster turnover rates, and this incredible thermal stability. It's really the best of both worlds, entirely new structures that are then fine-tuned by evolutionary pressure.
All right, part 4, building artificial enzymes, the ultimate hybrids.
Central to all of this cutting-edge research are artificial metalloenzymes, or ARMs for short. ARMs are pretty much the ultimate hybrid catalysts. They're built by taking a biomolecular protein scaffold and physically merging it with an unnatural transition metal complex. By embedding these unnatural metal cofactors right into a protein's pocket, scientists can essentially force the protein to catalyze new-to-nature chemical reactions, reactions that no natural enzyme could ever pull off.
And this hands chemists an entirely new toolkit to play with. Think about it. Instead of just relying on the standard 20 amino acids that nature gave us, scientists are now inserting new, non-canonical amino acids directly into proteins. They're binding noble metals, things like gold and rhodium, right into these pockets and using copper complexes for advanced biocatalysis. We are literally inventing new letters to add to the chemical alphabet of life. It's completely wild.
The breakthroughs we're seeing with these ARMs are nothing short of remarkable. Researchers are exploiting these highly flexible protein scaffolds and achieving synergistic catalysis, which basically means two different unnatural sites working together in harmony. But honestly, perhaps the biggest breakthrough is achieving greater than 99% enantioselectivity. Now, getting the correct 3D handedness of a molecule is notoriously difficult, but it is absolutely vital if you want to create safe drugs. And these artificial enzymes, they're doing it with near-perfect precision.
Which brings us to our final section, the future of chemistry, a green alternative.
Now, why does all this matter to you and me? Well, as the noble scientific background highlights, directed evolution and artificial enzymes offer a critically important green alternative. Think about traditional industrial chemistry for a second. It relies on harsh, toxic heavy metals, extreme temperatures, and just massive swimming pools worth of organic solvents. Engineered biocatalysts let us replace those toxic processes with mild, sustainable biotechnology. We are drastically lowering the amount of chemical waste and side products that get pumped into our environment.
And the best part? These aren't just pie-in-the-sky theoretical ideas. These are real-world applications happening right now. We're using evolved enzymes for the sustainable production of advanced biofuels like isobutanol to help replace fossil fuels. We're synthesizing complex, high-value pharmaceutical precursors with incredibly clean precision. We're even using whole-cell biocatalysis, which is basically turning living E. coli bacteria into microscopic, self-assembling chemical factories that pump out these designer molecules directly inside the cell. It's a total game changer. Which leaves us with this final thought. We've gone from simply observing nature's catalysts to actively directing their evolution, and now we're designing completely artificial, hybrid enzymes from scratch to perform reactions biology never even dreamed of. We're no longer boxed in by what nature requires for survival. Really, we're only limited by our own chemical imagination. So, what new-to-nature chemistry will humans dream up next? Thanks for joining me on this explainer, and keep questioning the limits of what's possible.